Hello everyone, this is Christopher Jackson. I'm going to be talking about elevated expression of NEU1 Sialidase promotes pulmonary fibrosis. The title of the article that I looked into is Elevated Expression of NEU1 Sialidase in Idiopathic Pulmonary Fibrosis Provokes Pulmonary Collagen Deposition, Lymphocytosis, and Fibrosis. The article was published in the American Physiological Society in 2016 and was conducted at the Baltimore Medical Center and University of Maryland School of Medicine. There's some background information that you all need to know in order to really have a better understanding of what's going on in this study. Fibrosis is defined as the thickening and scarring of connective tissue. Idiopathic relates to a disease or condition that has an unknown cause. Interstitial lung disease, abbreviated ILD, is a collection of diseases characterized by inflammation and fibrosis of lung tissue. It affects the interstitium of the lungs, which supports the alveoli. Interstitial lung diseases can develop due to genetics, exposure to hazardous breathing conditions, immune system problems, as a complication of systemic and pulmonary diseases, and can even develop for unknown reasons. Pulmonary fibrosis, it replaces functional lung parenchyma with scar tissue, which distorts the lungs and a patient's ability to breathe effectively. Basically, people who have pulmonary fibrosis are going to have trouble getting the necessary amounts of oxygen into their body. Pulmonary fibrosis affects about 140,000 Americans, and it's most likely to affect people aged 50 to 75. There's no known cure for pulmonary fibrosis. Idiopathic pulmonary fibrosis, abbreviated IPF, is the most common form of ILD. On average, patients survive only 2 to 3 years after being diagnosed with idiopathic pulmonary fibrosis. Despite new understandings of ILDs and the proteins involved in the diseases, the role of glycans, or saccharide chains, are still not well known. Sialylation is sialic acid residues which occupy the outermost portions of these glycan chains. They influence protein tertiary conformation and are terminally positioned on these chains, and they influence intermolecular and cell-to-cell interactions. Influenza recognizes the sialic acid residues within our lungs, and they use it to their advantage in order to know what to bind to so that they can infect us. Neuramidases, or sialidases, abbreviated NEUs, they hydrolyze the glycosidic linkage between sialic acids and their underlying subterminal sugars on the glycan chains. Changes in the sialylation state of glycans bound to proteins, or glycoproteins, may mask or unmask cryptic binding sites, and as a result will have functional consequences of the tissue. So why is all of this information important? Increased NEU activity was reported previously in bronchoalveolar lavage fluid from patients with interstitial pulmonary fibrosis. All that you really need to understand from that is that the fluid within the lungs contained more NEU activity. There's a relationship between the two. Inhibition of extracellular NEU activity reversibly inhibited fibroblast proliferation. There are 4 NEUs that have been identified in humans today, and NEU1 has been established as the predominant sialidase in airway epithelia and lung microvascular endothelia, which is why the study is going to be looking into the effects that NEU1 has on the lungs. Epidermal growth factor receptors, abbreviated EGFR, contributes to ILD pathogenesis. And the stimulation of these EGFRs increased NEU1 association with and desialylation with this receptor. It doesn't really go into much more detail about these epidermal growth factor receptors, but it has a relationship with NEU1 and the pathogenesis of these ILDs. Basically, this study is looking further into the role of NEU1 in the pathogenesis of interstitial pulmonary fibrosis, with an attempt to become closer in understanding the development and cellular mechanisms of interstitial lung diseases. The materials and methods section of this article went into a lot of complex and descriptive detail of how they performed all of the procedures in studying the effect that NEU1 has on lung tissue, but I'm just giving you the basic understanding of certain things that were done within the experiment. They used human lung tissue samples, cultured human lung cells, and also in vivo experiments in mice. I'm not going to talk at all about the in vivo experiments though. Normal, healthy lung tissue was used as a control and was compared with lung tissue samples from patients with interstitial pulmonary fibrosis. Pulmonary cells that were examined in this study were endothelial cells, epithelial cells, and fibroblasts of lung tissue samples, and they were examined in relation to being introduced to NEU1. Again, bronchoalveolar lavage. The point of this procedure is to examine cells from lung tissue of living individuals in order to compare the differences of healthy individuals and individuals with ILDs. The results of the study. NEU1 expression is higher in pulmonary cells, epithelial and endothelial cells, fibroblasts, from patients with IPF than in healthy controls. NEU1 delivery to endothelial cells caused substantial changes in gene expression, elevation and adhesion of T lymphocytes to endothelial monolayers, and also disrupted the endothelial cell capillary-like tube formation. Basically, NEU1 disrupted the normal endothelial cell function within the lungs. NEU1 upregulates collagen, mRNA, and protein expression in cultured normal human lung fibroblasts. The fibroblasts produce more collagen, suggesting that NEU1 contributes to collagen deposition in the lungs. Fibrosis. But surprisingly, it appeared during the experimentation that NEU1 upregulated collagen levels by attenuating or weakening collagen turnover rather than stimulating the production of collagen, which I thought was interesting because it was expected that it would stimulate the production of collagen before depositing it into the lungs, making it scarred tissue. So towards the end of the article, I found this figure showing photomicrographs of lung sections from control subjects, which were healthy individuals, and patients with interstitial pulmonary fibrosis. The blue was the staining for the NEU1, and figure A is just a control. It doesn't really need to be looked into as far as what I'm going to be talking about. The control or the healthy photomicrographs are shown in B, D, and G. The middle row shows bronchial epithelium in which IPF patients have increased NEU1 in the brush border of epithelial cells. You can see that by the dark staining in E and F with arrows pointing to where you should be looking. The bottom row shows endothelial cells in which IPF patients have increased NEU1 in endothelial cells. Again, look at the dark staining in figures H and I and compare them where these dark stains are in with the healthy controls. It's pretty obvious that NEU1 expression is higher in pulmonary cells from patients with IPF than in healthy controls. So basically, the study has given more insight on the mechanisms for interstitial lung diseases in relation to the role that NEU1 plays in the development of idiopathic pulmonary fibrosis. This study has also given scientists motives as to why future studies will need to be conducted in regards to the evidence that NEU1 plays a significant role in ILDs. If we can understand the mechanisms for certain diseases, we can begin to develop different methods of treatment to counteract the underlying cause to a disease.